And welcome to the session on first part of molecular genetics. I am Dr. Anju and I work at the Department of Medical Genetics, Kasturba Medical College, Manipal. So the objective of today's session is to learn about the applications of targeted molecular tests, especially those are PCR based and single sequencing based. So let us brush up the basics a bit. We all are made up of trillions of cells, and inside these cells, there is nucleus, which contains the genetic material. This genetic material appears in the condensed form as chromosomes, and we know that these chromosomes are made up of a linear molecule called the DNA or the deoxyribonucleic acid. So the DNA is a helical molecule and it is made up of nucleotide sequences. These nucleotides contain a pentose sugar, they contain a phosphate group, and they contain a carbon-nitrogen ring structure called a nitrogenous base, which is of four type, that is adenine, guanine, thymine, and cytosine. So this DNA, linear DNA molecule that has several functional units that is called gene. And this is just a representation of a gene. Although there are 19 to 20,000 genes, I am just showing a representation. Say this is an HBB gene, which has 3 exons. So these regions which are marked in green, they are called the exons, and they are the regions which ultimately would translate into a protein. In between these exons are the intervening sequences called the introns, which will be spliced out when a mature mRNA is formed. And before and after this gene, there are several regions like promoters, enhancers, and then the 3' nuclear region, which is responsible not only for the splicing of this or the process of transcription, but it is also responsible for regulation of the gene. That is, how much of the gene product will be formed, when will it be formed, is determined mainly by these regulatory regions, which are before and after a gene. So we know that any two unrelated human beings, they share 99.5% of the human genome. And there are several differences among individuals as well, which are genetically determined. So these changes can range from a single nucleotide to alterations of an entire chromosome. But we know that most of these changes, they just impact various traits, that is the color, the height, and so many other benign traits or the behavior. However, few changes, they might alter the normal protein and hence the bodily functions, and they are referred earlier, they were referred to as mutations. Now, preferably, we call them as disease-causing variations or pathogenic variations. So this is a one class of human genetic variation, which is called as a polymorphism. Polymorphism, by definition, is that change which is present in more than 1% of the allele in that particular population. And because they are common, they are usually inconsequential to the functioning of any gene. And that is why they are called as benign variants. They can lead to variant proteins, but not not detrimental to the protein function. And whenever they are present within the protein or the exons, they can result in different traits because they would result in the production of a parent protein. Other class of variations is the pathogenic or the disease-causing variation. So there are several classes of it. They can range from single nucleotide variation, which can be in the form of substitution of one base by the other. There can be deletion of few base pairs. There can be insertion of few base pairs. And the second category is that of indels, which are changes which are less than 50 base pair long. If there are more than 50 base pair long, they are referred to as copy number variation. And there can be larger chromosomal segment deletions, duplications, insertions, etc. They are all referred to as chromosomal rearrangements. So because of these detrimental changes, there can be several kind of disorders which have a genetic component. So if there are deletions, duplications, or there is change in number of chromosome, we refer to them as chromosomal or genomic mutations. If it is submicroscopic, it is often referred to as microdeletion and microduplication syndrome. Then if there are changes in just one gene, they are called single gene disorders, which can sometimes be CNVs, and most of the time they are single nucleotide variants. Sometimes the variations are there in the form of repeat expansions, for example, triplet repeat disorders. And sometimes the variations can be there within the mitochondrial genome, which is referred to as mitochondrial disorders. Sometimes the changes can be beyond the DNA sequence, and then they are referred to as epigenetic disorders, one example being imprinting disorders. And often there is a component of both genetic as well as environmental factors, and these are referred to as complex disorders. So how do we choose which test to be done for these disorders? There are some basic principles. Whenever we have a clinical diagnosis and there are very few variants which are known to cause that disease, we can check for only those variants. I will be discussing the examples a little later. And then when we have a diagnosis and the target area is very small, say maybe few exons, few variants in a gene or a small gene, again, it can be done through a targeted testing. And sometimes whenever the genomic architecture, for example, tandem repeats are there and the mutation involves these repeat regions, they may have to be investigated using a targeted test. Now, often in the clinic, we are using broad spectrum or genomic tests like panels, exome sequencing, genome sequencing, and these are usually employed whenever the target region is large, whenever a condition is caused by more than one gene, or if we are not able to make a clinical diagnosis, we have to go for these unbiased broad spectrum tests. So we are going to learn few of the PCR-based methods and their techniques and principles as well. So this is how a polymerase chain reaction works. We all know that it's a DNA cloning method. DNA cloning method means it is used to amplify a small amount of DNA into large quantity of DNA. So it takes a specific sequence of DNA in small amount and amplifies it so that it can be used for further testing, and hence it is the basis of almost all the molecular tests. It is quite rapid, it's extremely sensitive and robust as well. So what all do we have to add for the PCR to work? We should have a DNA template, that is our region of interest, which we want to amplify. Then there are particular primer sequences which will be complementary to the DNA template. We need an enzyme so that DNA synthesis can occur. And for DNA synthesis, we need these nucleotides or the DNTPs of all 4 subtypes. And then we need few more chemicals for the reaction to occur. So this is what happens inside a thermocycler or a PCR machine. Once we put our genomic DNA, the other chemicals and the primers, and we apply heat, this double-stranded DNA gets resolved into single-stranded molecule. And once it is single-stranded, the primer will come and bind. Both the forward and reverse primers come and bind, which leads to synthesis of another strand of DNA. And then there is another cycle of heating, which will again separate this double-stranded molecule into 2 single strand, and again the same cycle is repeated. So for after 30 to 35 cycles, there will be huge amount of DNA that we will get from a small quantity of DNA with which we started. So there are some modifications of these PCR that has been used for investigating some genetic disorders. One of these is allele-specific PCR or amplification refractory mutation system, in short also known as ARMS PCR. So ARMS PCR till a decade ago was very common test that was used for detecting a lot of single nucleotide variants. So the principle of ARMS PCR or allele-specific PCR is a requirement for precise base matching between a 3' end of a PCR primer and the target DNA. So here we can see that there is a wild type allele. So the primer at the 3' end, which has the sequence of a wild type allele, will go and match, and then it will amplify. However, if there is a mutation, when you put this primer, which will have a mismatch at this 5' end, there won't be any amplification. So this is the principle of allele-specific PCR. So this test has been widely used for detecting common mutation in HBB gene, which can be multiplexed and they can be investigated at once. However, this test has now been replaced largely by sequencing of the HBB gene, which turns out to be more cost effective and is able to detect most of the mutations that are present in the HBB gene. Another modification of PCR is PCR RFLP. Here the principle is that of restriction fragment length polymorphism. So with the help of several restriction enzymes which recognize particular sites in the genome and they cut through it, there will be different fragments of different lengths that will be produced if we use these restriction enzyme. So how do we determine which restriction enzyme to choose depends on the type of mutation. So we need to be sure that whatever enzyme we are using, it has to encompass the wild type and the mutant allele. So then only we will be able to appreciate the difference. So PCR RFLP is till now used as one of the common test and a very robust and simple test for detection of a common disorder, that is spinal muscular atrophy. There are 4 clinical subtypes of spinal muscular atrophy, although type 1, which is the infantile form, it is a very common cause of a floppy infant or a floppy baby and starts at almost 2 to 3 months of age. So here, a very common mutation is the deletion of exon 7 of SMN1 gene, which is present in as many as 95 to 98% of cases with SMA. And it can be readily picked up with the help of PCR RFLP, where we can detect the deletion of exon 7. Another class of disorders where we need definitely a targeted test is that of repeat mutations or dynamic mutations. And these mutations, they lead to very complex architecture, which is often not amenable to PCR and Sanger sequencing. So this is an example of one of the disorders where we can use PCR for screening whether somebody has this disorder or not. So this is an 8-year-old male that we had seen in our clinic. There was family history of 2 maternal uncles being affected with similar intellectual disability, autism spectrum disorder, and few facial dysmorphic features. So our first clinical diagnosis was that of a Fragile X syndrome. So we have done a PCR screening here, and what happens in Fragile X syndrome is that there are usually more than 200 CGG repeats in an affected individual. And when we do PCR to detect this region, you will see that if there is no expansion of the allele, you will see the PCR product is there. So in control 1, control 2, control 3, and control 4, which are of normal individuals, we can see a PCR product. However, because of the expanded repeat size of more than 200 in individuals who are affected with Fragile X syndrome, there is no product that is seen here, which means that there is a possibility of Fragile X syndrome. However, this has to be validated and confirmed by another line of testing. The next technique that we are going to talk about today is Sanger sequencing. Sanger sequencing method was developed by Frederick Sanger, after whom it is named back in 1977. This is also known as di-deoxy chain termination method because it uses the principle of deoxy and di-deoxy nucleotides for DNA synthesis. So here we can see that this is a ribose sugar. This is a phosphate group attached to it. And in this ribose sugar, there is a deoxy, there's a hydroxyl group here. And this is what is going to interact with the phosphate group and lead to DNA synthesis. However, in di-deoxynucleotide, there is only a hydrogen molecule which is there. And this would interrupt the chain and there won't be any DNA synthesis happening here. So what we do in case of Sanger sequencing is, in the reaction mixture, we put primer, we put DNA template, we put DNTPs, how we had put for PCR. There is a polymerase for the DNA synthesis to occur. And then we put fluorescently labeled di-deoxynucleotides of all 4 subtypes. So whenever the chain is getting elongated and it encounters a di-deoxynucleotide, it gets terminated. And then it is run through a capillary sequencer, and then we come to know about the sequence of the target region that we were investigating. The Sanger sequencing is still considered gold standard for sequencing. And even if we investigate any individual with broad spectrum testing or NGS-based testing, still we need to validate the sequence with the help of Sanger sequencing for confirmation. So we will see how we have used targeted Sanger sequencing in few of the families that we have come across in the clinic. So this is a 1-year-old male child. The parents complained of short stature, and on examination, he had a short stature which was at -6.7 st. There was frontal bossing, depressed nasal bridge, mid-face protrusion was there, and along with short uh, arms, he also had rhizomelia. On skeletal survey, he had shortening of all long bones. So we made a clinical diagnosis of achondroplasia. In achondroplasia, the single mutation at this position, that is C1138, it causes 99% of the cases of achondroplasia. So it is quite simple to test once we have a clinical diagnosis. So this principle of having recurrent variants can be used if a very good clinical diagnosis is possible, and hence it becomes a very cost-effective method for getting a definitive molecular diagnosis in these families. This is another family of a 3-year-old male baby, and there was history of recurrent fever since the boy was 5 months of age. He also was diagnosed with hemolytic anemia and had received 5 blood transfusions. He had organomegaly, and the HB electrophoresis showed very high levels of HbF, that is around 93.7%. So clinically, we made a diagnosis of beta thalassemia major. Although for the diagnosis, these investigations were enough, but this was the first child in the family, and they were looking for more reproductive options. Hence, we asked them or offered them mutation analysis or genetic testing so that they will be able to undergo prenatal diagnosis in the subsequent pregnancy. So this is what we found on Sanger sequencing. We found there was an intronic variant, which is a common mutation in the Indian population. Another common mutation was there in exon 1. So in the proband, there were 2 mutations, one C92 plus 5 G to C and another C92 G to C. And then we can see that one of these variants is inherited from mother. She did not carry the other variant, and father had wild type allele for this mutation, however, was heterozygous carrier for the second mutation. And this is what we call as a compound heterozygous variant. That means 2 mutations have been inherited from one parent each in this child, which confirmed the diagnosis of beta thalassemia major. This is the last example. This is a 3-year-old girl. She's born out of consanguineous marriage. She was well till 2 years of age and then had neuroregression, spasticity, and seizure, which were all progressive. And we asked for an MRI because of so many neurological issues. And when we looked at the MRI, we could see that there were ventricular white matter abnormalities of the deep cerebral white matter. And there was thyroid appearance as well. Taken together, it was very suggestive of metachromatic leukodystrophy. And the gene for metachromatic leukodystrophy is aryl sulfatase A or ARSA. So we did Sanger sequencing since it is a small gene, and we found that there were 2 parents, which again were coming from one parent each. So with this, I would just like to highlight that whenever possible, a targeted testing using several PCR-based methods or Sanger sequencing can be done to achieve a definitive molecular diagnosis in individuals with genetic disorders. And this is very helpful for their appropriate management and their prognostication, as well as to discuss the natural history with the family. This also helps us to give them informed genetic counseling, again, regarding the risk of recurrence, the at-risk members in the family, and the risk of recurrence in the subsequent children, and offering them prenatal diagnosis, which is not possible if there is no molecular diagnosis available. It also helps in carrier testing for extended family members as well. Thank you very much.